Welcome, everyone. Today, we're breaking down a major systematic review and meta-analysis that tackles a really critical question. Can we use non-invasive brain stimulation, or NIBs, to bring some real relief for those incredibly challenging non-motor symptoms of Parkinson's disease? Things like cognitive decline and depression. Let's dive into the evidence and see what it tells us. So here's how we're going to break it down. We'll start with the background, why this research is so needed in the first place. Then we'll look at the methods they used to sift through all the evidence. After that, we'll get to the good stuff, the core results for both cognitive and neuropsychiatric symptoms. And just as importantly, we'll talk about the limitations of the evidence before we get to that final clinical take-home message. All right, let's get right into it. First things first, why does this research even matter so much? What's the fundamental problem that scientists are trying to solve here? This slide really lays it all out, doesn't it? On the one hand, you have the immense clinical burden of non-motor symptoms. We're talking about everything from cognitive decline to debilitating depression. These are often what impact quality of life the most. And on the other hand, you have the rationale for NIBs. The whole idea is that these symptoms might come from dysfunctional brain networks. And maybe, just maybe, we can use these techniques to reach in and sort of retune those networks. So how did the researchers go about gathering and filtering all the evidence? A big review like this is only as strong as the studies it includes, so their method for choosing them is absolutely crucial. Let's take a look. Okay, take a look at this. This flowchart really shows you just how picky the researchers had to be. They started with, get this, more than 11,000 potential articles. And through a really careful, step-by-step screening process, they whittled that massive number all the way down to just 34 high-quality, randomized controlled trials that perfectly fit what they were looking for. And how did they stay so focused? Well, they used this super strict framework called PICO. The population had to be adults with Parkinson's. The intervention had to be repeated sessions of NIBS. The comparison, and this is key, had to be against a sham or placebo control group. And the outcomes they measured were changes in those non-motor symptoms. Having these strict rules is what gives a study like this its power. All right, this is what we've been waiting for. The results. Let's start with what they found when they looked at the impact of NIBS on cognitive symptoms. Now, before we jump into the numbers, there's a really critical distinction we have to get our heads around. The researchers didn't just lump all the studies together. They split them up based on whether the stimulation was excitatory, think of it like trying to turn up the activity in a brain region, or inhibitory, which is like trying to turn it down. This separation is fundamental to understanding everything that follows. So first up, let's look at those excitatory or turn it up protocols. The researchers pooled all the data from studies that use things like high-frequency TMS on brain areas like the prefrontal cortex. And the finding? Well, frankly, it was negative. They found no significant improvement in global cognition, attention, memory, or executive function when compared to the sham treatment, not right after treatment and not at follow-up either. But then, they looked at the inhibitory or turn-it-down protocols, and this is where things get really, really interesting. And here we go. The meta-analysis found that inhibitory stimulation, specifically low-frequency TMS targeting the right side of the prefrontal cortex, was significantly better than sham for improving global cognition. And the really impressive part? This wasn't just a flash in the pan. The effect was still there, sustained at the 3-month follow-up. That suggests a potentially lasting benefit. And just look at that p-value, less than 0.00001. Statistically speaking, that is a home run. It indicates a very strong finding. But, and this is a huge but that the authors are very quick to point out, this incredibly promising result is drawn from only 2 studies. So while it's a powerful signal, it's one that absolutely needs to be confirmed in bigger trials before we can get too excited. Okay, so that's cognition. Now let's switch gears and turn to the neuropsychiatric symptoms. Things like depression and anxiety, which, as we know, are just a major source of disability and reduced quality of life for people with Parkinson's. When it came to depression, the story got a bit more complicated. For the excitatory stimulation, targeting areas like the left prefrontal cortex, there was a significant benefit, but it was short-lived. The effect was gone by the 1-month follow-up. In contrast, that inhibitory protocol, targeting the right prefrontal cortex, sound familiar? Once again, showed an effect that was better than sham and was actually sustained at 1 month. Anxiety followed a pretty similar pattern to what we saw with depression. There was a significant short-term benefit from excitatory stimulation, but again, that effect didn't last. Meanwhile, for apathy, which is another incredibly challenging symptom, the news wasn't as good. The studies unfortunately showed negative findings right across the board, with no significant benefit compared to sham. It's also worth quickly mentioning two other areas the review covered. First, a different type of brain stimulation called TDCS, or transcranial direct current stimulation, generally showed negative results for both cognition and depression. And second, for atypical Parkinsonism, that includes conditions like Lewy body dementia, the data was just way too limited, making it impossible to draw any real conclusions for those groups. So we've got some promising signals, right? A few glimmers of hope. But before we get carried away, we have to do what all good scientists do. We have to talk about the limitations, because the quality of the evidence matters just as much as the results themselves. And yeah, this chart tells a pretty clear story. The authors rated the overall certainty of the evidence as low to moderate. As you can see, the vast majority of the 34 trials they included had either some concerns or even a high risk of bias. Now, this doesn't mean the findings are wrong, but it's a huge piece of context. It means we have to interpret everything with a healthy dose of caution. So what were some of these flaws? Well, for example, many studies didn't properly account for people who dropped out of the trial, which can really skew the results. Other common issues included problems with how patients were randomized into groups and studies deviating from their original plans, all things that can weaken how much we can rely on the final conclusions. And this is a fascinating detail. They found that some of those positive results we talked about were, well, fragile. They did what's called a sensitivity analysis. And for some outcomes, like anxiety, if you took just one specific study out of the mix, the statistically significant result would just vanish. It really shows you how much a single trial can influence the overall picture, and it hammers home the need for more, larger studies. So this all leads to a really important question, one you might be asking yourself right now. Why the split results? Why did cognition generally respond so poorly, while symptoms like depression and anxiety seem to do a bit better, at least in the short term? Well, the authors suggest a pretty plausible biological reason. Think about it. Cognitive decline in Parkinson's is a widespread problem. The wiring issues are kind of all over the brain. So trying to fix that huge diffuse problem by stimulating one small spot on the surface is probably just not enough. In contrast, for something like depression, we have much more established targets, like the left prefrontal cortex. That might be why stimulating that one spot can lead to more consistent, even if they're modest, effects. Okay, let's bring it all home. What's the final clinical bottom line from all of this? And maybe even more importantly, what has to happen next in this field to get us clearer answers? The great thing about this paper is it doesn't just point out the problems, it offers solutions. The authors lay out a fantastic, clear, 4-step roadmap for future trials. This includes things like using brain imaging to pick better, more personalized targets, designing much larger trials with enough statistical power, fine-tuning the stimulation protocols themselves, and figuring out which types of patients are most likely to respond in the first place. So, what is the clinical bottom line? Let's be clear. For cognition, the current data does not support the routine use of knives in Parkinson's. For mood, there's some exploratory evidence for short-term benefits, but these findings have to be interpreted with extreme caution because of all those methodological issues. Ultimately, the future of this therapy isn't in a one-size-fits-all approach. It's in a big shift toward personalized, brain connectivity-guided treatments. The clear message is that we desperately need more rigorous, high-quality research before these very promising techniques can become a standard of care for patients.